where we're looking at the preparative and the payoff and the overall net gain. And again, keep in mind, any of these could be radio labeled. Any of these could be then transitioned through the metabolic process that we're doing and providing a positional labeling as well as a concentration. So again, let's start with glucose. If I had 6 millimoles of glucose and I'm going to get hexokinase to do its particular job, how many millimoles of ATP am I going to be consuming to produce 6 millimoles of glucose 6 phosphate? Think about it. We're going to be needing the same ratio of glucose to that of ATP consumption. The enzyme is going to do its job. You might need a very small amount of enzyme because it's going to be basically consuming and producing the product from the substrate, and it'll keep doing that time and time again. So the enzyme concentration is not so much important. It might change the rate of reaction, as we understand with our kinetic process, but the enzyme itself still requires a matching amount of ATP because that phosphorylational point is going to have to match the concentration of the phosphorylational point on the glucose. So again, if I had 6 millimolar of glucose, I would need 6 millimoles of ATP. I'd produce 6 millimoles of ADP, and I'd produce 6 millimoles of glucose 6 phosphate. It's that simple, law of mass conservation. And remember, if I had 6 millimoles of this particular reaction and my delta G prime 0 was equal to minus 16.7 kilojoules per mole, I can work out how much delta G do I need for or produce with 6 millimoles of glucose. So think about that, okay? And again, you should be able to do this math. This is coming from your general chemistry and your organic chemistry and now applying it to biochemistry, okay? It's just not issue about structures and modifications and pathways. There are definite quantificational aspects in regards to this particular system. So we've now got this glucose molecule, and we've now phosphorylated it in position number 6, and we've consumed ATP in the process utilizing an enzyme. It has taken us minus 16.7 kilojoules to do this, so that's really good. It's going to give us a negative delta G, so it's going to be in moving from the reactant to the product side. So when we look at our process of these 4 steps, and again, think about any of these particular carbon positions could be radio labeled. The phosphate could be P32. The hydrogens could be tritiated in regards to having their own label. But again, those positions are going to be equated to very specific positioning and retention of those positions in the molecule. And this is why we illustrate this particular process in regards to the colorations here. So we identify carbons 1, 2, 3, 4, and 5, and 6 on our molecule of glucose. We consume ATP to make the glucose 6-phosphate by using the enzyme hexokinase. This is a commitment stage. It's only in one direction. When we see in our metabolic pathways a reversible direction, it means it can go either way. But these steps number 1 and step number 3 are commitment stages to the metabolic process. So we're going from glucose, consuming ATP, and making glucose 6-phosphate. So again, here's the 6th position. We've got glucose there. We then use the phosphohexokinase isomerase to cause it become from a 6-membered to a 5-membered ring, but still conserving all the number of elements and carbons and everything, hydrogens and oxygens there, to produce fructose 6-phosphate. Okay. And again, we've identified the carbon number 6 and the carbon number 1, but carbon number 1 in fructose 6-phosphate is not phosphorylated until it goes through the phosphofructokinase number 1. I've got to be very careful how I say that because that's a tongue tire. But that process also is a commitment stage in regards to consuming ATP. So in that process, we're utilizing ATP and to provide that phosphate component to the number 1 carbon position to then produce the fructose 1,6-bisphosphate. Okay, so we've got our 2 priming reactions in regards to producing now the fructose 1,6-bisphosphate. So go back to my lecture on the tutorial on radioactivity. We are now going to go through and utilize an enzyme called aldolase, a reversible enzyme, taking the fructose 1,6-bisphosphate and cleaving it in this position. So the first part, the dihydroxyacetone phosphate, is going to conserve carbons 1, 2, and 3. So remember, 6 carbon moiety of glucose, and then 6 carbon moiety of fructose 6-phosphate, 6 carbon moiety of fructose 1,6-bisphosphate. But once it gets to that aldolase, we are going to degrade it and produce 3 carbon constituents, or 2 moieties of a 3 carbon constituent. Okay, one is going to be glyceraldehyde 3-phosphate, one is going to be dihydroxyacetone phosphate. But again, we use another enzyme which will take that dihydroxyacetone phosphate and bring it back to glyceraldehyde 3-phosphate, increasing the concentration. And this is where we're going to now get the payoff stage, because we've invested 2 moieties of ATP to get to this fructose 1,6-bisphosphate. So in this case, carbons 1, 2, and 3 produce the dihydroxyacetone phosphate, and carbons 4, 5, and 6 produce the glyceraldehyde 3-phosphate. So if I had a radio-labeled position in position number 4, so let's have a look. It's carbons number 1, 2, 3, and 4. So we had this guy radio-labeled. But as you can see, what happens with that position number 4, it's here in the beta position. And then we're going to see it shift into the 5-fructan component from the prion to the fructan in regards to the 5-membered ring, and giving us a new position, but it's still retaining its radioactivity. And if we have that same one in radioactivity land, and we're going to go through then the aldolase reaction to cleave the 5-membered ring to produce the 2 3-carbon moieties, that radio label is going to be retained specifically in the glyceraldehyde 3-phosphate. But we're going to have to be very careful where it's going to be positioned. So where is the radio label going to be positioned here? So let's have a look. This is carbon number 6. Is carbon number 6. Is carbon number 5. Carbon number 5 is carbon number 4. Carbon number 4. So in that guy, that would be the radio labeled position if carbon number 4 was radio labeled. Okay, so hopefully this is clear, because again, it will be on the exam in one way, shape, or form in radio label tracing molecules from glucose all the way down to the issue of producing pyruvate, which is going to be now our next stage of the payoff. But we still have to understand some of the systems that are occurring enzymatically. So let's have a closer look at what's occurring. So we have glucose 6-phosphate being produced by the hexokinase at the consumption of ATP. This is an enzyme system. Substrate comes in, produces the glucose 6-phosphate, and that glucose 6-phosphate can have 2 fates. And that can go into the pentose phosphate pathway, which we'll cover very shortly in, I think, most probably next lecture or on next Tuesday. I think next lecture, actually. And we're going to have an amount of particular energy required to produce this particular system. So to go from fructose 6-phosphate to glucose 6-phosphate to fructose 6-phosphate, it's going to require us to have a positive 1.7 kilojoules per mole. And again, this is an enzyme-mediated situation. That particular position gives us the capacity to control and regulate, because without that positive energy, we are not going to have that reaction occur, because it's always going to favor the glucose 6-phosphate unless we feed it energy through having a coupled reaction. Now, with the fructose, with the phosphofructokinase number 1, we are now going to consume an additional ATP and producing inorganic phosphate and ADP to give us the dual labeled position of the phosphorylation occurring in this yellow part here. Now, this particular reaction is very favorable, negative 14.2 kilojoules per mole. And in this case, we will produce the fructose 1,6-bisphosphate, as we've seen previously in the other diagram. Now, this is a commitment stage, because we cannot reverse, again, the issue of the arrows, understanding where the reversibility and where the single arrows are very key components in glycolysis and in gluconeogenesis, as well as in the TCA cycle. So in this case, we'll produce the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, as we see in the mauve and the blue regions here. But this particular reaction also is a regulatory reaction because it's going to contain and require 23.8 kilojoules per mole to move through. Okay, so again, we have the capacity to regulate this by prohibiting the amount of energy that is needed to move this reaction forward. So this represents the preparative stage in regards to the system. But I want to focus a little bit on the issue of the fructose 1,6-bisphosphate producing the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, which is illustrated on page 540. And basically, we have our enzyme cleft. Aldolase is very well known and structurally determined, and how the fructose 1,6-bisphosphate works with that particular system. It goes through a number of particular mechanisms interacting with certain acid bases which represent amino acids. So again, these represent amino acids, particularly like this lysine here, and a basic amino acid and an acidic amino acid, to give this orientation reaction the best way to move through and degrade the molecule and change it to a new form. I don't expect you to know this and off by heart, but I want you to appreciate, importantly, we're still dealing with three-dimensional structure. We're still dealing with enzymes and amino acids in those enzymes to interact with a substrate, in this case, that of fructose 1,6-bisphosphate, similar to what we saw when we saw with the degradation of an aromatic containing amino acid peptide chain in chymotrypsin. So we can go through the whole process in regards to see how we see the first release. So there's a sequential release, like again we saw with the chymotrypsin. There is going to be binding of the substrate to the enzyme and a sequential release. And the first release in this case is the glyceraldehyde 3-phosphate. Because what we're going to have to do is regenerate the cleft of the enzyme with the appropriate charge states and the appropriate amino acids that have interacted with the substrate to allow us then cause an energy destabilization. Remember, we talked about the issue of the transitional state in enzymology. We want to cause that destabilization and allow then the final product to actually be released from the enzyme cleft with all the regeneration of the charge states of the amino acids that are originally there before the substrate came in. And again, this is the cyclization of what we see with enzyme activity. So in this case, we see glyceraldehyde 3-phosphate being the first product to be released. We still have the remnants of the individual substrate still being there, being worked on by the various amino acids in that enzyme cleft. And finally, we get hydrolysis, which will allow us utilization of molecular water, again, how molecular water plays a reactive species within this particular system, which will then cause the release finally of the dihydroxyacetone phosphate. So we see this sequential release, again, what we see and saw similarly in the chymotrypsin system that we use as an example, and then ideally the restoration of the enzyme cleft with the various base amino acids, the lysine being protonated, this particular amino acid having the protonation and ready to accept the next fructose 1,6-bisphosphate to come in and do the work. And remember, this is happening in milliseconds in regards to basically the kinetic rate and reactions and the specificity of the reactions. So again, we're looking at compounds that inhibit this particular process or competitive binders. Remember, we've talked about competitive inhibitors. It's going to slow down the rate of the reaction. It's going to slow down the enzyme function and the production of glyceraldehyde and 3-phosphate and dihydroxyacetone phosphate. But again, allosteric modulation of this enzyme, so we have the cleft, but any allosteric modulation of that enzyme which causes a structural change in the enzyme cleft might also perturb the rate of kinetics, meaning decrease the kinetic rate, or might actually even destroy the kinetic rate because the substrate can no longer bind to the enzymatic cleft. So again, thinking about our issue of three-dimensional structure and how this plays a role with allosteric modulation and competitive inhibition, or even uncompetitive inhibition. So again, all the concepts we've talked about in enzymology, with the Birkeland-Weaver plot, as well as the Michaelis-Menten equations, come into play when we look at these metabolic pathways. So again, that's why we did those first off, because if I was trying to talk to you now and talk about all this stuff, you would have no idea what I'm talking about. But that's why we've done that sequential learning process. So let's recap. We've come back to our glyceraldehyde 3-phosphate and our dihydroxyacetone phosphate. Now, that's been our investment stage. We've had to invest ATP into this. So how are we going to make sure we have the energy balance? Because remember, it's an overall energy reaction in this particular pathway. And now we're going to move into the payoff phase of the system. So we have the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate playing a particular role. And remember, we're now going to have the triose phosphate isomerase interconvert the dihydroxyacetone phosphate to become glyceraldehyde 3-phosphate, because the payoff stage here requires and only consumes glyceraldehyde 3-phosphate. It does not consume dihydroxyacetone phosphate. We have to have that isomerase to bring it into that particular form. So think about this issue like we talked in the tutorial regarding radioactivity. If one of these particular molecules was radioactive carbon, that means we can have two types of molecules of glyceraldehyde 3-phosphate. And they're going to have to go through separate processes, because at the final product, one is going to be radio labeled and the other is not. And they're going to have the conserved position of that carbon-14 being present in that particular molecule. So there's going to be some of those in the homework. There can be some of those in the tutorial, which you will actually in the recitation. So let's now look specifically at that payoff phase. How are we going to make sure that this reaction moves forward? Because we've consumed ATP in our preparative stage. So we need two molecules of ATP to make a balance. But as you now will understand, we are going to produce a lot more ATP, because instead of having one molecule of glyceraldehyde 3-phosphate, a 3-carbon constituent, we have dihydroxyacetone phosphate through the triose phosphate isomerase, which is now going to produce two molecules of glyceraldehyde 3-phosphate, this guy here. So with that interconversion, we have now two molecules of glyceraldehyde 3-phosphate, which are going to go through the whole metabolic process and give us the payoff. So let's go through it. So two molecules of glyceraldehyde 3-phosphate are going to go through the glyceraldehyde 3-phosphate dehydrogenase. So when we see this word dehydrogenase, it means we're going to have an electron transfer. In this case, we're going to make NADH. So we're going to be sinking electrons and producing protons, and we're going to have those safely contained in this particular constituent to be later used elsewhere. And we'll see how that is utilized in coming lectures. So we're going to consume two molecules of inorganic phosphate. We're going to consume two molecules of the reduced NAD+, and this is a reversible reaction. Again, it's not a commitment state, it's a reversible reaction. So in this case, we're now going to produce two molecules of 1,3-bisphosphoglycerate. So again, we have this capacity to now phosphorylate this particular molecule utilizing the glyceraldehyde 3-phosphate. So we consumed two molecules of phosphate, and the electron sink the NAD+ to give us this position and phosphorylate it from the aldehyde to the carboxylic acid. Okay, so we now have glyceraldehyde, 2,3-bisphosphate glycerate, which is now going to be a very viable molecule to cause the release of this phosphate group from this particular ester to produce now ATP. Remember, we have two molecules of this coming through. So we're going to consume two molecules of ADP and make two molecules of ATP utilizing the enzyme, which is reversible, the phosphoglycerate kinase. Okay, so a kinase causing the removal of that phosphate group. We then produce the 3-phosphoglycerate molecule. That 3-phosphoglycerate is going to go through a mutational mutasal reaction, causing a rearrangement through the phosphoglycerate mutase reaction. Again, a reversible reaction. And so we're going to see this particular phosphate group move over to this position here, and this become a hydroxyl, and this become a phosphoester bond. In that particular position, this guy is more susceptible to leaving, but ideally we're going to try and reduce this particular guy first off by utilizing the loss of water, because we're going to form a double bond here, and that is going to happen through an enzyme called enolase, which again is reversible, by taking this time the 2-phosphoglycerate, so we've gone from 3-phosphoglycerate to 2-phosphoglycerate, releasing molecular water and producing then 2 moieties of phosphoenol pyruvate. Now, the phosphoenol pyruvate is going to be a great energy source for us, because once we get that phosphate bond gone, we're going to have a completely dephosphorylated molecule in the form of pyruvate kinase, in the form of pyruvate via the pyruvate kinase. In that particular process, it's a one-directional commitment stage. So we have a one-directional, it's not reversible. And in this case, we're producing 2 ATPs because we're consuming 2 ADPs. So we have 2 reactions that are producing overall 4 moieties of ATP. That is how we're going to get the payoff stage. But also, we've produced 2 moieties of NADH. And those NADH, in some cases, are worth more than the ATP, as we'll see a little later on. So you're going to have to understand, be able to draw this, be able to trace potential carbon positions within this molecule using a radio-labeled tracer. And you're going to be able to also give me concentrations. So if I started off with, in this case, 5 millimoles of glucose, I'm going to end up with 10 millimoles of pyruvate, because I've produced 2 equivalents of glyceraldehyde 3-phosphate because of the triose phosphate isomerase to allow us to get off into the payoff stage. Hopefully this is clear. You're going to have to understand this, and as well as the points of control. We will come back to these points of control shortly. Maybe it's actually the next slide. So let's have a look what's going on with the reaction. Glyceraldehyde 3-phosphate, the phosphate, 2 per glucose molecule, and therefore we're going to get 2 overall of NADH being consumed. But again, it's happening within an enzyme. The NAD plus is sitting there ready to accept those electrons. We're going to have a histidine and a cysteine being coordinated in regards to allowing the substrate, the glyceraldehyde 3-phosphate, to actually first bind to the cysteine and coordinate with the histidine to move those particular electrons and allow us then to transfer those electrons and those protons to the NADH. And in this case, we're going to see positive 3.6 kilojoules per mole to get to that particular reaction to produce the 1,3-bisphosphoglycerate. We're going to then consume ATP to make ATP. So this is going to be a great reaction in regards to producing an energy carrying currency in the form of 3-phosphoglycerate. This particular reaction with the delta G prime 0 is negative 18.5 kilojoules per mole. So again, this is the first part of our particular reaction. So we're kind of getting to the stage where we're coming ahead of ourselves, because at this stage, we actually account for the consumption of the ATP in regards to the preparative stage that was allowing us to produce the glyceraldehyde 3-phosphate and the dehydroxyacetone phosphate. But we're going to take it one further step. We're going to take the 3-phosphoglycerate and go through a mutase reaction and shift that particular guy around to allow us to produce the 2-phosphoglycerate. Again, a small energy requirement, so a potential regulatory component in regards to 4.4 kilojoules per mole. We then have the 2-phosphoglycerate. Again, we've swapped the positions of the phosphate, and this is what happens here. We actually get a biphosphorylated version, and we'll get a favoritism of one going to the other in causing that shift of that phosphate group into this position, as we see with the 2-phosphoglycerate. So at the moment, the phosphate group is in position 3. We want to put it into position 2. With that 2-phosphoglycerate being present, we have the enolase, which is going to be a dehydration reaction. But again, it requires positive 7.5 kilojoules per mole. We then get to the issue of having the phosphoenol pyruvate. Now, the phosphoenol pyruvate, as I said, is this massive energy carrier. Once we get removing that phosphate group and rearrange that double bond, as we see here, to produce a double bond in this position with the release of that phosphate group, which is going to be consumed by ADP to make ATP, we're going to see negative 31.4 kilojoules per mole. So a whopping amount of energy coming from that reaction. In this case, we then produce pyruvate, which is our key component to the next enzyme pathway in the TCA cycle, which we'll get to in about, I think, next week after the exam. So we have these transformations, the degradation of glucose, the phosphorylation of ATP, and the transfer to produce NADH from NAD plus consumption. So again, we have this issue of these tautomerizational situations where basically tautomerization gives a greater capacity to have reactivity, as I spoke to you yesterday. So in this case, we have all these little positive components within the metabolic pathway. But look at, again, the amount of energy that we will get from one single reaction, which will drive these smaller positive reactions. But without this being happening, we are not going to be able to couple these reactions and move them forward. So again, the phosphoenol pyruvate and the utilization of ADP with magnesium, with the pyruvate kinase, gives us capacity to produce that ATP, which we're going to utilize to pay off our debt in the preparative stage and produce enormous amount of energy in regards to negative 31.4 kilojoules per mole, which will then also promote the small increments of positive delta G prime 0 that we need to continue this reaction to move forward. So let's have a look at the overall action in regards to summation of the reaction. We have glucose, which will require the consumption of 2 NAD plus, 2 ATP, 2 pyruvate, 2 inorganic phosphates. That is going to ultimately give us 2 moieties of pyruvate, 2 moieties of NADH, 2 protons, 2 ATPs, 2 water molecules. So let's have a look at the half reactions. So again, the half reactions, glucose and 2 moieties of NAD plus will give us 2 pyruvates, 2 NADH, and 2 protons. And remember, these guys we haven't taken into consideration of the amount of energy they're going to produce. We've taken consideration of that these will produce energy, but we're going to process these a little better. But also we still have pyruvate, and we're going to further degrade pyruvate to extract more energy out of that particular molecule. So we're only going for a first part of the reaction. We're going to get more energy as we degrade that pyruvate. But let's see what we just did in the issue of glycolysis. Now we have ADP and PI producing inorganic phosphate. Producing ATP and molecular water. And again, we've got to think about the issue of the comparative summation and the right orientation of those delta G prime zeros. So we have glucose on the left-hand side, we have pyruvate on the right-hand side. So therefore, the orientation of this particular reaction is correct. Now we have ADP and PI. I've actually switched this over already. So normally it would be ATP, molecular oxygen, going to ADP and PI at negative 61 kilojoules. But I've already switched it over for convenience. So we see it's going to consume basically a positive 61 kilojoules per mole. But again, from that first part of the reaction, the summation of the delta G prime 0, so we have delta G1s, delta G2s, and then summation of the delta G. Remember, delta G1, 2 are summated. We we're going to see minus 85 kilojoules per mole. But again, that is the first part of the reaction of glycolysis. We now have a molecule called pyruvate, a 3-carbon constituent, which we're going to further degrade to carbon dioxide and to water, completely metabolize that. By breaking those bonds, we're going to get more energy. And that is going to be through the citric acid cycle and oxidative phosphorylation. And we'll be studying those in coming lectures. I'm just getting you through the issue of being able to understand the first stages of degradation of glucose. So let's think about glucose itself. So we have glucose, 10 consecutive steps in the overall process to produce pyruvate. Now, with pyruvate, if we go through anaerobic conditions in regards to no oxygen being present, we're going to produce lactic acid, and that's going to stay there until we can replenish the system with oxygen and allow it to become pyruvate again. Pyruvate itself can also go through more anaerobic conditions in regards to utilizing microorganisms to produce ethanol. So we can use yeast in this case to take pyruvate, producing carbon dioxide, as well as ethanol. Okay. In this case, when we take pyruvate down our mainstream, so our mainstream, when we go through aerobic conditions, we're going to produce carbon dioxide, and the pyruvate is going to be degraded to form acetyl-CoA. We've already been introduced to acetyl-CoA, and that acetyl-CoA is going to be the key entry component into the TCA cycle and the Krebs cycle. When we see that going in, we're going to fully degrade this to carbon dioxide and water. And with that, we're going to actually extract a lot more FADH2, NADH, and ATP equivalent. We're going to produce actually GTP. Remember, we had ping pong, where we can take the GTP and transfer the base to the phosphate to make from ADP, and phosphate position from GTP to make ATP. So we'll see that a little later on. So again, we're going to make a lot of energy from this system if we're going down this particular component. But we've got to think about also this issue of energy overall. So if we think about glucose consumption and the changes with molecular oxygen being available, we can get 30 to 32 ATPs. Now, I put that issue of 30 to 32 because we haven't considered the issue of transportation, because there is a situation where we will consume 2 ATPs due to a priming reaction. We'll talk about that a little later on. But can you see how much energy we lose if we are in an anaerobic environment? That we only produce 2 ATPs because we're either going in this direction or that direction in regards to producing a byproduct of ethanol, which is still highly containing carbon, oxygen, and hydrogen, and can't be degraded any further. And it's actually, in some cases, toxic to the cells. So enzymatically, we see a differentiation based on the presence of molecular oxygen. We'll talk about that a little later on when we look at the interface of the metabolic pathways going from the glycolysis to the TCA cycle. Enzymatically, we have identified a couple of enzyme systems which are only in one direction, and that is the kinase, the phosphofructokinase, and the pyruvate kinase. That should be pyruvate kinase. Now, the interesting thing is we also have the issue of our transportation and our regulation from the hormonal regulation of the GCPRs in regards to insulin to increase gene expression. We've all seen how that insulin system works, how an epinephrine or noradrenaline gives that capacity to increase the movement of glucose into the cell by increasing the transporters and also promoting the enzymes that are going to handle the amount of glucose coming in. Those enzymes are the same enzymes we've now just studied in regards to glycolysis. So again, we have to have a matching situation. If we're bringing in more material to come into the enzyme system, we need to increase the enzyme concentration. So again, we're going to have to increase and regulate the expression of those particular proteins. And we can do that by steroidal hormones and as well as these secondary messenger systems, as we are now familiar with the cascades of the phosphokinase A, the inosol 3-phosphate system, as well as the regulation of GLUT1 and GLUT2 transporters. Okay, so we have to start thinking about our bigger picture of what we've studied in the past and bring it in now into a perspective of what we are doing. There's always going to be an equal response in regards to if I'm going to produce more glucose in and or consume more glucose, where am I going to store that? How am I going to store that? How am I going to transport it? And how am I going to metabolize it? So there's very key points of regulation in regards to this particular system. Here we have a nice little illustration of the whole thing that's going on in regards to our cell. So here's our situation of glycolysis. And remember, I told you about the glucose 6 phosphate can go to the pentose phosphate pathway, which we'll study a little later on, produce the ribose sugars, which we're going to be needing for the production of DNA and RNA. We have the transport system to increase the movement of glucose into the cell to feed this particular system. But that hexokinase becomes a very, very important regulator. We have also the capacity to inhibit glucose degradation, glycolysis, if we are basically too energetically favorable. Therefore, we might close down the insulin receptor and the IP3 system and the protein kinase B to inhibit the transport system and move those into the vesicles to stop the movement of particular carbohydrates into the cell because we can't handle them. But also what we see here, and this is where we're getting to our fatty acids a little later on, where the fatty acids, depending on how much glucose and how much energy the cell is going to need, it's going to depend on where and what happens with these particular fatty acids. And we're going to utilize these fatty acids as a supplemental source to keep fueling the mitochondria, because it's this mitochondria, the powerhouse of the system, that's going to give us approximately the 30 ATPs that we require to keep the cell happy for every single molecule of glucose. So if I'm going to consume 1 millimolar glucose, I'm going to produce approximately 30 millimoles of ATP. Okay, so this is the bigger picture I'm trying to get you to kind of be familiar with. We've gone through this. We've gone through the transport system. We've gone through glucose movement. We are now at this situation of understanding what happens with glucose. And we've now got down to the issue of being able to understand, importantly, the production of pyruvate. We're going to move in the future to oxidative phosphorylation in the mitochondria and the utilization of these fatty acids as an energy source, so as an alternative energy source. So we're going to talk about a little later on ketogenesis or ketoacidosis and beta-hydroxyacetone, beta-hydroxybutyrate and acetoacetate as alternative energy systems. But that's going to be a little later when we talk about metabolic diseases such as what we see with insulin and how that changes the metabolism of carbohydrates. So we've got a big picture now in regards to what's occurring with the multifaceted that we've talked about in the previous 8 weeks, and we're going to now move forward. Now, cancer is a problem. Cancer is basically used to be called the wasting disease because one of the early diagnoses of cancer was basically this massive decrease in glucose circulation. So what happens? Cancer is basically a cellular system that is in a slight anaerobic mode. Therefore, it must really get glucose as a primary energy source, producing lactic acid, which is a byproduct of certain anaerobic metabolism. So one of the old days, in the very, very old days, when they used to think you had cancer, they'd see an increase in lactic acid because what you'd be doing is increasing your consumption of carbohydrates, particularly glucose, but in a slightly anaerobic environment because of the way the tumors are formed in the tissue. We also see in cancers where basically the enzymes start switching to increase the enzymes present in the glycolytic pathway. And that was another basic biochemical test, which is still used today in regards to the concentration of those particular enzymes in circulation. Because again, you're promoting the feeding of a cancerous material, which is rapidly dividing. It's not going through the normal phase clock of cellular reproduction. So think of a clock face, cellular reproduction, 12 o'clock, 1 o'clock, 2 o'clock, 3 o'clock, 4 o'clock, etc., where we start with replication and at 11:59, basically it's cell death, apoptosis. But what we see with cancer cells, they go from the 12 o'clock position to the 7 o'clock position, then they go back to the 12 o'clock position. They never go through the apoptotic or cell death or programmed cell death. But in the same process, they are promoting the enzymes that they need to ensure that they have the capacity to survive. They cannibalize the tissues around themselves to get the nutrients that they need because they don't have all the mechanisms. And in this case, they can starve healthy tissues of both the oxygen and the nutritional values, the carbohydrates, because they are basically consuming those tissues to feed that rapid division of cells. And that's why cancer is called the wasting disease, because this is exactly what happens. We see an invagination, increased anaerobic environment, circular depletion, increased glucose concentrations being required by the cellular material, increased glycolytic pathway enzymes, and basically tissue damage surrounding the tumor. So again, this is an area of oncology or cancer research which you can get involved with if you're doing research or want to go into a graduate program. There's some undergraduate biology, cell molecular biology programs that might encroach on cancer biology. So again, they cannibalize their tissues to do this. So let's have a look at what also occurs in regards to some of the situations versus hypoxia. Hypoxia is an environment where there's a low oxygen environment. And this could be through suffocation. It could be restriction of airways. And see what happens in regards to this particular system. So we have glucose uptake in cancer. We see increasing the transport system and increasing the enzymes in regards to the cancerous form, as we talked about before. We see a hypoxic environment occurring where the cells become more more anaerobic, where they're not getting the required oxygen. We then start seeing some other issues occurring in regards to glycolytic enzymes being further increased, and starting to see an increase of certain particular byproducts being produced, such as glucose 6-phosphate and glycerol 3-phosphate and pyruvate, which is our normal metabolic pathway. But we might start seeing 6-phosphogluconate by the dehydrogenase system in regards to being produced, because this guy is slowing down. We're trying to get all that ATP, and with the hypoxia, we're going to have a serious problem of not producing the required ATP to feed the cells. So again, we're going to have some problems occurring. We have cancer also impacting aerobic respiration within the mitochondria by basically starving it of the required nutrients to allow you to go through oxidative phosphorylation. So again, with cancer, we see the change in the transport system, changing in the enzyme system, metabolic byproducts which could be very greatly increased, which are cancer markers in regards to being able to determine what is occurring in that particular system. So again, we have some metabolic pathways or metabolic systems that are not normally apparent in a cancer cell, but in a normal cell, but become apparent in a cancer cell. And therefore, these guys become indicators of particular diseases such as cancer. But again, it's this continual fight between the aerobic and anaerobic, and how glycolysis is going to try and feed those particular systems by providing the ATP. Okay. So glucose itself is not the only carbohydrate that gets into the system of glycolysis that we've seen today. We have a number of different sugars that have the capacity to work with that particular system. We have galactose, which is commonly found in milk, goes through a system in regards to its transformation of galactose to become glucose to enter glucose 1-phosphate. So we didn't see glucose 1-phosphate, we saw glucose going to glucose 6-phosphate automatically. But we have a potential capacity to allow glucose to enter as glucose 1-phosphate. This could be from glycogen, starch, dietary starch, or how we've stored our glycogen. And we're going to use a phosphorylase to do that to produce the glucose 1-phosphate. That glucose 1-phosphate can then go through the glucose phosphate phosphoglucomutase and switch that phosphate from position number 1 to position number 6. Then it becomes a substrate quite clearly for the hexokinase and go through the normal processes of going through the fructose 6-phosphate, fructose 1,6-bisphosphate, and glyceraldehyde 3-phosphate, etc., and dihydroxyacetone phosphate. We also see mannose, which is another sugar, 6-mannose. Basically, mannose is a carbohydrate. It can be phosphorylated by a kinase, consuming ATP, to produce mannose 6-phosphate. But it also then uses an isomerase to isomerize mannose to produce the fructose 6-phosphate, which then can directly feed into the glycolytic pathway. We have sucrose, which is table sugar, and we can digest that with a sucrase and basically produce two moieties of carbohydrates, one being glucose and one being fructose. The glucose can go through a hexokinase, like we see normally with the glycolytic pathway, consuming ATP to produce the glucose 6-phosphate, which will then feed the normal pathway. With the resulting fructose, we can use another kinase, hexokinase, consuming ATP to give us the fructose 6-phosphate. So again, we can phosphorylate this particular position in the 6th position and feed it into the glycolytic pathway. So I'm illustrating here how other sugars come into the metabolic pathway, the central pathway of glycolysis. We have galactose, mannose, glucose, fructose, and sucrose. We have a number of other particular sugars. We have galactose, and we have threulose through different enzymes going through the production, in this case, of D-glucose. Now, fructose is an interesting one because it can be divided into two forms. It can also go through the whole process and allow us to actually produce dihydroxyacetone phosphate and glyceraldehyde and dihydroxyacetone phosphate. But noticing this is not the glyceraldehyde 3-phosphate. That comes later on with a triose kinase. So again, from fructose, we can utilize a fructokinase to label this in the number 1 position. So the number 1 position is here in regards to having fructose 1-bisphosphate, and then using a fructose 1-phosphate aldolase to cause that cleavage, which will give us the carbons 1, 2, and 3 to give us a dihydroxyacetone phosphate, which we can now feed into the normal system using the triose phosphate to make glyceraldehyde 3-phosphate. But we have the resulting molecule of glycerate, so not labeled, so this guy is not labeled in the form of glycerate. That glycerate can be then, again, phosphorylated using a kinase, a triose kinase, and from glyceraldehyde produce glyceraldehyde 3-phosphate. So there's a number of alternative pathways that we can bring sugars in to our particular metabolic pathway of our central form of our preparative stage and our payoff stage, more so in our preparative stage in forming the glyceraldehyde 3-phosphate, which will then feed into the payoff stage of making that NADH and those ATP molecules and releasing finally a moiety, a 3-carbon moiety of pyruvate. We are going to further study this particular guy and its degradation to produce more energy. So again, we haven't finished our degradation of glucose yet. So I want to have a look at a few systems here in regards to carbohydrates. We're going to talk about this tomorrow in regards to looking at starch molecules and how we degrade these particular starch molecules to give us the carbohydrate moiety, such as glucose 1-phosphate, and then make it into glucose 6-phosphate. But we're going to have to figure out, importantly, what we're going to do with those particular points of entry and how enzymes play a particular role. So I will leave it at that in regards to looking at now reducing sugars and how we're going to metabolize those reducing sugars to feed our glycolytic pathway. Okay, we'll take it from there, and I'll open for office hours now. Okay, so hopefully it's come to